Imagine you're watching a toddler, right? They're playing in the dirt, having a great time. They reach for a shiny rock, and bam, they get this tiny, almost invisible paper cut.
Happens all the time.
Exactly. And within milliseconds, just beneath the surface of that skin, this invisible, microscopic war begins. I mean, millions of cells are already communicating. They're deploying chemical weapons and making these literal life or death decisions about what belongs in the body and what needs to be, destroyed.
And all of this happens without you ever feeling a single thing.
Yeah. But it makes you think, what happens when that system makes a mistake? Like when the weapons turn on your own tissues.
Well, it's a terrifying prospect, honestly, but it highlights just how finely tuned this machinery has to be. The immune system is basically constantly walking this tightrope between aggressive defense on one side and peaceful tolerance on the other.
Right.
And to really understand how it manages that balance, you have to look past the surface level definitions. You really have to dig into the actual mechanisms.
And for those of you listening who are prepping for boards, or maybe you're trying to make sense of some new clinical trial for an immunotherapy drug, you know that immunology is so often treated like a black box.
Oh, absolutely.
Because you spend years studying anatomy where everything has a physical place, right? And physiology makes sense because it's just pumps and pipes.
Yeah, it's very mechanical.
But then you hit immunology and suddenly you're drowning in this massive alphabet soup nomenclature. It's all interleukins, CD markers, and these cascading pathways that seem to come out of nowhere.
We hear that frustration constantly from medical learners. I mean, you're asked to memorize these transient chemical signals without actually understanding the spatial or logical context of, well, why they even exist.
Okay, let's unpack this. Our mission today is to break that black box wide open. We are doing a deep dive using the Illustrated Dictionary of Immunology, 3rd edition by Julius Cruz and Robert Lewis.
It's a fantastic resource.
It is, but it's massive. It's a very comprehensive text. So we're going to distill it down into a clear, logical framework for you. We want to open beyond just memorizing acronyms to really understanding how these cells communicate, the weaponry they deploy.
And what happens when the blueprints are flawed?
Exactly. Plus, how modern immunopharmacology is hacking these exact processes.
So we need to start with the grand architecture of defense. The text divides this into 2 primary limbs, which are innate and adaptive immunity.
Right, the classic division.
Yeah, but it's crucial to understand that these aren't just 2 sets of tools. They represent 2 entirely different evolutionary strategies. Innate immunity is your natural, native defense. It's present from birth.
And it's pretty generic, right?
Right. It relies on recognizing broad molecular patterns, things that are just generally dangerous, like bacterial cell wall components.
I always think of it like a nightclub bouncer. The mechanical barriers, your skin, the mucosal surfaces, the sheer acidity of your stomach, that's the velvet rope.
I love that analogy.
Yeah, if you don't have an ID, you just don't get in.
And if a troublemaker manages to slip past that velvet rope, say, through that paper cut we talked about, the innate cellular responders take over. These are your macrophages, your polymorph neutrophils, or PMNs, and natural killer cells.
The security guards inside the club.
Exactly. They're already inside, programmed to physically toss out anyone acting aggressively.
But I've always wondered about the limitation there. Because the innate system is incredibly fast, but it seems, really blunt. Like if you get infected with a novel virus, the bouncer doesn't know who they're looking for.
And that is the fundamental limitation. Innate immunity lacks immunological memory and precise specificity.
It just sees trouble and attacks.
Right. It holds the line immediately, which keeps you alive today, but it doesn't adapt for tomorrow. And that is where the adaptive immune system comes in. This is the highly specific response mediated by your T and B lymphocytes.
The heavy machinery.
Yeah, it features clonal selection, precise self versus non-self recognition, and crucially, long-lasting memory.
So if the innate system is the bouncer, the adaptive system is like a specialized detective agency. They take a few days to build a highly specific profile on this sophisticated cat burglar.
Right.
But once they have that profile, they never forget the face. 10 years later, if that burglar tries to break in, the detectives are already waiting at the window.
What's fascinating here is how the biology manages the kinetics of these 2 branches. The adaptive primary response has a distinct lag period.
Because it takes time to build the profile.
Exactly. When those naive T and B cells encounter a specific antigen for the first time, it takes several days for them to metabolically ramp up, proliferate into an army of clones, and differentiate into effector cells.
But the second time is different.
Totally different. Because they leave behind a specialized pool of memory cells, a second encounter with that exact same antigen triggers a secondary response that is just massive and rapid. The lag time practically vanishes.
Wait, okay, so if the innate bouncer is at the site of the paper cut in the finger, and the adaptive detectives are waiting in the lymph nodes, how does the bouncer hand off the suspect's profile?
Ah, the handoff.
Yeah, because they can't just wander around the bloodstream hoping to bump into each other. That would be like finding a needle in a haystack.
You've hit on arguably the most critical event in immunology, which is antigen presentation. This handoff requires a highly coordinated commute. It's executed by what we call professional antigen-presenting cells, or APCs.
Right, the messengers.
Right. And this elite group includes dendritic cells, macrophages, and B cells. Their job is to capture exogenous antigens at the site of infection, process them internally by chopping them up into smaller peptides, and then display those peptides on their cell surface.
The text mentions they display them on the major histocompatibility complex, or MHC class II molecule.
Yes, class 2 is key here.
But I always struggle to understand why the APC doesn't just, spit the chopped up antigen back out into the blood for the T cells to find. Why does it need this specific MHC display case?
Well, because freely floating debris could accidentally trigger a massive immune response in the wrong place at the wrong time. T cells are actually deliberately programmed to be blind to free-floating antigens.
Oh, wow. I didn't realize they were literally blind to it.
Yeah. They will only recognize an antigen if it's physically held in the molecular grip of an MHC molecule on the surface of a professional APC. It's a biological failsafe. It basically proves to the T cell that this antigen was processed by a trusted sentinel.
That makes so much sense. So the dendritic cell has the evidence in its MHC display case. Now it needs to find the T cell. Which brings us to the physical anatomy of the immune system.
Right. Location matters.
The textbook details how lymphocytes mature in primary lymphoid organs. So T cells in the thymus, B cells in the bone marrow. But they don't stay there.
No, they migrate to the secondary lymphoid organs, places like the lymph nodes, the spleen, and mucosa-associated lymphoid tissues like Peyer's patches.
So those secondary organs are essentially highly organized meeting hubs.
Exactly. Think of a dendritic cell acting as a roaming sentinel in your skin. We call them Langerhans cells when they're in the skin.
Okay, Langerhans cells.
Right. After it captures the pathogen from our toddler's paper cut, it physically changes. It stops hunting, detaches from the surrounding tissue, and literally crawls into the lymphatic vessels. It flows like a river into the draining lymph node.
It's commuting to the local police precinct.
And that lymph node is packed with millions of naive T and B cells. They are constantly circulating, just looking for their specific match. When that dendritic cell arrives, bearing the antigen on its MHC class II tray, it scans through the T cells until it finds the one single clone with a matching receptor.
That is wild.
Dendritic cells are unparalleled in this role. They're the most potent activators of naive T cells.
So once that match is made, the alarm sounds. The naive T and B cells are activated, and we shift from surveillance into the effector phase, which means we need to look at the arsenal they deploy. Yeah. And the adaptive arsenal is split into two main branches. You have humoral immunity, driven by B cells, and cell-mediated immunity, driven by T cells. Let's focus on humoral first, because this solves a massive logistical problem.
It really does. Because if a pathogen is circulating everywhere in the blood, you need a weapon that can travel everywhere.
Right. So when B cells activate, they differentiate into plasma cells, which are essentially microscopic antibody factories. They just pump out immunoglobulins, or IgA.
And those immunoglobulins are highly specialized based on the geographic needs of the body. For example, IgM is the first responder.
The big clunky one.
Yeah, exactly. It's a massive pentameric molecule, meaning it has 5 units linked together. It is somewhat clunky, but because it has 10 binding sites, it is excellent at grabbing onto pathogens early in the infection before the body has time to really refine its response.
And then you transition to IgG, which is the heavy hitter. It's a smaller, sleeker molecule, making it the main protective antibody in your serum.
And if you're a medical learner listening to this, circle this fact. IgG is the only antibody class that crosses the placenta to protect a developing fetus.
Yes. The textbook explains it does this via a very specific transport mechanism called the bramble receptor, or FcRn.
Crucial receptor.
It physically binds the IgG on the maternal side
Yeah.
and actively ferries it across the placental barrier into the fetal circulation.
And that active transport is just vital for neonatal survival. Now, moving to the mucosal surfaces, so the gut, the respiratory tract, you have IgA.
Mucosal sentinel.
Right. If IgG is the heavy hitter in the blood, IgA is what stops a bacteria from setting up camp in the mucosal lining, where blood concentrations are much lower. It literally bathes the epithelial surfaces, neutralizing pathogens before they can even attach.
And finally, there's IgE,
Mm,
which binds tightly to mast cells and serves as the trigger for allergic responses and defense against parasitic worms.
exactly.
Okay, so what about the T cells, the cell-mediated weapons?
Well, T cells take a much more direct cellular approach. You have your CD4 positive helper T cells. These are basically the generals of the immune army.
Okay, so they give the orders.
Right. For instance, the Th1 subset secretes a cytokine called interferon gamma. This signal hyperactivates macrophages. So if a normal macrophage is a standard security guard, an interferon gamma-stimulated macrophage is like a microscopic terminator.
Wow.
It aggressively destroys even the most stubborn intracellular bacteria. And then you have the CD8 positive cytotoxic T cells. Their job is to seek out our own host cells that have been infected by a virus and force them to undergo apoptosis or programmed cell death.
They destroy the factory so the virus can't replicate.
Exactly.
But we can't talk about the arsenal without mentioning the complement system. The text places this right at the bridge between innate and humoral immunity. It's this cascading series of proteins circulating in your blood, labeled C1 through C9.
Yeah, but I think the word cascade almost glosses over how violent this actually is.
Oh, it's incredibly violent.
A cascade means biological amplification. When C1 is activated, perhaps by an IgG antibody bound to a bacteria, it acts as an enzyme to cleave and activate multiple molecules of the next protein in the chain.
So one C1 activates 10 C2s.
Right, which activate 100 C3s and so on. It is an avalanche of proteolytic cleavage that culminates in multiple proteins assembling into the membrane attack complex, or MAC.
And the MAC physically punches a pore right through the bacterial cell wall. The bacteria's internal contents leak out, and it bursts.
It's brutal.
Here's where it gets really interesting, though, because when we talk about antibodies, a lot of people just assume the antibody itself acts like a poison dart that kills the bacteria.
Oh, that is such a pervasive misconception. Antibodies are essentially just highly specific protein labels. They rarely kill bacteria directly on their own.
They need help.
Yeah, they rely on other systems like the complement cascade we just described or phagocytic cells to actually do the killing.
And the text highlights this mechanism called opsonization. Antibodies act as opsonins. The authors state that opsonization makes microbes, quote, delectable to phagocytes.
I love that phrasing.
It's a brilliant analogy. Because some bacteria have these slippery, sugary capsules that make it incredibly hard for a neutrophil's receptors to get a grip on them. But when that bacteria gets coated in IgG antibodies and complement proteins, you've essentially given the neutrophil a molecular handle to grab.
Exactly.
It's like putting hot sauce on a bland, slippery meal. The phagocyte just gobbles it up instantly.
It's a beautifully coordinated lethal machine. The specificity of the antibody directs the brute force of the macrophage.
But biology is messy. We have this lethal military machine perfectly tuned. What happens when the blueprints are flawed from birth or, if the targeting system glitches?
Right, and this brings us to immunopathology. We can broadly categorize these system failures into two buckets. First, you have immunodeficiencies, where the system fails to mount an adequate defense. And second, hypersensitivities and autoimmunity, where the system is overactive or directed against the host itself. Let's look at deficiencies first. These can be primary, meaning genetic, or congenital, or secondary, meaning acquired due to environmental factors.
Under primary immunodeficiencies, severe combined immunodeficiency, or SCID, is just a devastating example.
It really is.
In SCID, a genetic defect wipes out both functional T and B cells. So these infants are completely defenseless against opportunistic infections. Because they lack the entire adaptive limb, it's usually fatal in infancy without a bone marrow transplant to replace those defective stem cells.
And on the other end of the primary spectrum, you have selective IgA deficiency. This is actually the most common primary immunodeficiency.
The mucosal one.
Exactly. Because these patients specifically lack mucosal IgA, the barrier function of their respiratory and gastrointestinal tracts is compromised.
So they get sick more often.
Right, it leads to recurrent pulmonary and GI infections, even though their systemic IgG in the blood might be perfectly normal.
Then you have secondary immunodeficiencies, which represent a massive global health burden. HIV/AIDS is the most famous, specifically infecting and destroying the CD4 helper T cells.
Removing the generals from the immune army?
Precisely. But you also see it with aging immunosenescence. The textbook explains this begins with the natural involution or shrinking of the thymus over time.
Less thymic tissue means less naive T cell output.
Right, which forces the elderly to rely on a progressively narrower pool of memory cells.
But if you're working in global health, the text highlights a critical, often overlooked fact. The most frequent cause of secondary immunodeficiency worldwide is actually malnutrition.
That's a huge point.
Immune cells have massive metabolic demands. When a naive T cell is activated, it needs to undergo rapid clonal expansion. If the patient is suffering from protein energy malnutrition, the body simply lacks the amino acid building blocks required for that cellular proliferation.
So the system just stalls out.
Yeah, T cell function plummets and complement activity drops drastically.
So that's the first bucket. The second bucket of system failures is autoimmunity. This is a failure of self-tolerance. During development in the thymus and bone marrow, the immune system is supposed to learn to ignore your own tissues.
Right, the education phase.
But when that education fails, you get diseases like type 1 diabetes, where T cells and macrophages aggressively destroy the insulin-producing pancreatic islets, or Hashimoto's thyroiditis, where lymphocytic infiltration destroys the thyroid follicles.
And sometimes the damage is just collateral, like an immune complex disease, which is a type 3 hypersensitivity. Antigen-antibody complexes form in the blood and get physically lodged in the microvasculature.
Break in the kidneys.
Exactly, like the tiny renal glomeruli. They get stuck there and inappropriately activate the complement cascade. The resulting avalanche of inflammation destroys the surrounding healthy kidney tissue.
I always struggle to understand how the body usually prevents this. Like, if a T cell receptor accidentally binds to a self-antigen on a normal cell, isn't that enough to start a war? Why does the body require more than just that initial recognition?
If we connect this to the bigger picture, this is exactly why the text emphasizes the Brescher-Kahn theory of two-signal activation. It is the core failsafe against autoimmunity.
Okay, two signals.
Right. The theory states that for a naive lymphocyte to become fully activated, signal 1, which is the antigen binding to the T cell receptor, is not enough. In fact, if a T cell receives signal 1 alone, it induces a state of permanent unresponsiveness called anergy. The cell effectively turns off.
Oh, so it assumes it made a mistake.
Precisely. To fully activate, the T cell also requires signal 2, a co-stimulatory signal. And here is the crucial part. Signal 2 can only be provided by a professional antigen-presenting cell that has also been activated by innate danger signals.
So it's a two-key system.
Exactly. It ensures that T cells aren't just reacting to random self-proteins they bump into, but are only reacting to proteins presented by an APC that is actively confirming, yes, there is an infection here. When this two-signal requirement is bypassed or glitches, autoimmune disease takes root.
And understanding that failsafe isn't just, academic trivia. It is the literal foundation of modern medical intervention. We aren't just using broad-spectrum steroids anymore to indiscriminately hammer the immune system into submission. We are precisely hacking the communication lines.
It's incredible how far we've come.
Let's look at how we manipulate these processes, starting with vaccines.
Vaccines are the induction of active immunity. Edward Jenner laid the groundwork using the cowpox virus to safely immunize against smallpox. He didn't know about T cell receptors, of course, but he was exploiting the fact that the two viruses shared identical surface epitopes, creating cross-reactive immune memory.
But modern engineering takes this so much further. The text discusses conjugate vaccines.
Oh, these are fascinating.
Right. A bacterial polysaccharide, which is a sugar chain from the bacterial capsule, usually only triggers a very weak, short-lived humoral response because it can't activate T cells.
Because T cells only recognize peptides, right?
Exactly. So immunologists chemically link that sugar to a highly immunogenic carrier protein. That protein recruits CD4 helper T cells into the response, tricking the immune system into mounting a massive T cell-dependent long-lasting memory response against a simple sugar molecule.
It is brilliant translational medicine, and that same engineering prowess gives us targeted immunotherapy, specifically the clinical use of monoclonal antibodies, or mAbs.
The famous mAbs.
Right. The text outlines how these are engineered using hybridomas, or advanced phage display libraries, to create synthetic antibodies that target highly specific human receptors.
This is where we need to slow down, because the mechanism for the drug abatacept is just mind-blowing if you understand the two-signal theory we just talked about.
Right.
Abatacept is used for severe rheumatoid arthritis. Walk us through how it works.
Sure. So abatacept is a recombinant fusion protein. It is specifically designed to target and bind to CD80 and CD86.
Okay, CD80 and 86.
Remember, CD80 and CD86 are the surface markers sitting on the antigen-presenting cell. They are the markers responsible for delivering that crucial signal 2 to the T cell.
Let's visualize this. If activating a T cell is like starting a car, signal 1, the antigen, is putting the key in the ignition.
Right.
But the car won't move unless you press the gas pedal.
Yeah.
Signal 2, delivered by CD80 and CD86 binding to CD28 on the T cell, is the foot pressing the gas pedal.
Exactly. What abatacept does is physically bind to CD80 and CD86 on the APC. It essentially puts a massive molecular boot over the gas pedal.
Wow.
So even if the T cell has the key in the ignition, even if it is recognizing the joint tissue and rheumatoid arthritis as an antigen, it cannot receive signal 2. The handshake is blocked.
And without signal 2, the T cell becomes anergic. You are literally flipping the off switch on the inflammation driving the arthritis.
Yeah.
We are rewriting the instructions. By understanding surface markers like CD4, CD8, CD28, we can tell the system to actively ignore a transplanted kidney, or flip it the other way and remove the brake so the immune system suddenly recognizes and attacks a stealthy tumor cell.
The vocabulary of immunology has basically become the programming language of modern therapeutics. And if we connect this to the bigger picture, it requires us to rethink the fundamental philosophies of the field. For decades, the textbook rule of immunology was the concept of self versus non-self discrimination. The idea that the body attacks anything foreign and tolerates anything that is self.
Right. That's what's taught in week 1 of medical school.
But the text introduces a really provocative paradigm shift. Polly Matzinger's danger theory.
Oh, that's good.
She proposed that the immune system doesn't actually care if something is foreign. It only cares if it is dangerous. It requires a danger signal, specific cellular damage or distress, to activate those antigen-presenting cells so they can provide signal 2. A foreign protein that enters the body but causes absolutely no tissue damage might be entirely ignored.
Wait, so if a surgeon cuts me open to give me a kidney transplant, the surgical incision itself is causing massive tissue damage. According to Matzinger, that physical damage provides the danger signal that wakes up the APCs.
Yes.
Is that why transplant rejection is so aggressive compared to just, say, a random foreign protein entering the gut?
That is exactly the debate her theory sparked. And it provides a fascinating lens for tumors as well.
How so?
Well, a tumor is technically self-tissue, so under the old model, the immune system should just ignore it. But a rapidly growing tumor causes local hypoxia and tissue destruction.
Which are danger signals.
Right, and the immune system does try to fight tumors, which supports Matzinger's theory. It really makes you ask, does this reframe how you view autoimmune diseases? Are they a failure of self-tolerance, or is the immune system responding to an invisible cellular distress signal we just haven't identified yet?
That completely flips the script on everything we just built up.
Yeah.
But, that's the beauty of this field. It's constantly evolving. Let's briefly recap our journey today. We started at the velvet rope with our innate bouncers.
The first line of defense?
We watched the dendritic cells capture evidence and commute to the lymph nodes to present it on their MHE trays. We explored the arsenal, the IgM first responders, the IgG heavy hitters, the IgA mucosal sentinels, and that violent complement cascade.
We also examined the pathologies of immunodeficiency and the two-signal failsafe of autoimmunity.
Right. And finally, we saw how drugs like abatacept literally hack that failsafe.
It's a vast amount of information, but it all connects through that underlying logic of surveillance, recognition, response, and memory.
It really does. And before you go, we want to challenge you, our medical learners, with a quick review exercise based on what we covered today, just to solidify your learning.
Get ready.
Test your recall. Imagine a 6-year-old patient presents to your clinic with recurrent sinopulmonary infections and gastrointestinal issues, specifically giardiasis. You run blood work, and it shows completely normal IgG and IgM levels.
Kernel systemic level.
Exactly. So what is the most likely primary immunodeficiency, and what specific mucosal antibody are they lacking? Think back to the specialized geographic functions of the different immunoglobulin classes we discussed.
If you followed the logic of the mucosal barriers and which antibody acts as the sentinel there, the diagnosis should lock right into place.
The answer is selective IgA deficiency. Thank you for joining us as we unpack this incredible text. The next time you encounter a wall of immunological jargon, remember, It isn't just a black box of random letters and numbers.
Not at all.
It's a highly organized, deeply logical detective agency, and you now have the master key to understand how it operates. Keep questioning, keep learning, and keep diving deep.